Hello and welcome back. In this session, I'm going to be taking up formation of male gametophyte and its structure. So by the end of this session, you're expected to achieve these learning goals. 1. Explain the 2 stages of a microspore maturation in a pollen grain. 2. Provide justification for the asymmetric pollen mitosis. 3. Describe the structure of a mature pollen grain. 4. Give a brief account on anther dehiscence. 5. Determine viability of some pollen grains. 6. State one advantage and one disadvantage of pollens to humans. Now, to start with, let us recall the transverse section of an anther. So we learned that in an anther, in a bilobed anther, we have 4 of these microsporangial sacs. And as the anther starts maturing, the sporogenous tissues inside these microsporangial sacs, which are diploid in nature, starts undergoing the maturation and differentiates into microspore mother cells. And from those microspore mother cells, we obtained haploid microspores by the process of meiosis. Now these haploid microspores, which are there carried in the microsporangia, would further undergo growth and differentiation. And in the process of the microspore maturation, once the microspore starts becoming unicellular, then we can be expecting the microspore to be undergoing certain changes in order to be forming or in order to be becoming into the male gametophyte or pollen grains. Now, in order to understand the process of microspore maturation, what I'm going to be doing is, for our convenience, let's consider a single microspore. Alright, so even though we can be expecting thousands of microspores in each of these microsporangia, but let me draw a single microspore. So let's say that this is a single haploid microspore. Alright, and this single haploid microspore so this is our haploid microspore. Now this starts undergoing maturation, and during the process of maturation we can be understanding that there are 2 distinct stages. So in the first stage, what happens is this haploid microspore would undergo into a process which is known as vacuolation. Okay, so in the first stage we can be speaking about a process which is called as vacuolation. Now, what do we mean by this process, vacuolation? Vacuolation is basically a process where we have the formation of a single large vacuole inside the microspore. Okay, and because of this formation of the large vacuole, the cell starts to expand, right, or the cell starts to enlarge. Okay, so we get let us see, the cell has expanded and it has formed a single large vacuole inside, and most of the central space is occupied by this large vacuole, and because of which the nucleus of the microspore starts to get pushed towards the wall. Right? So here is the nucleus and this is the large vacuole, and this process of the formation of a single large vacuole inside the microspore cell is the first stage, which is called as vacuolation. Now, in the second stage, after this formation of the vacuole, then the microspore further undergoes into another event, and this event is called as asymmetric mitotic division. Okay, so the second stage here is it undergoes into an asymmetric division, and the nature of this division is mitosis. Okay, so we commonly have a reference of to this mitosis as pollen mitosis, and this is the first pollen mitosis, and this first pollen mitosis is considered to be asymmetric. Right, now you will understand why this mitosis has to be called as an asymmetric mitotic division. The reason is because this microspore cell, right, this enlarged microspore, this is still the microspore, but it is larger in size. So this enlarged haploid microspore cells now forms two daughter cells as a result of mitotic division, right? But these two daughter cells are unequal in size, right? So we get to see inside the microspore, we get to see two daughter cells, right? But these two daughter cells are of different sizes. Right? You can be seeing a large daughter cell, and this larger daughter cell is known as the vegetative cell. It's called as vegetative cell, or it can be even called as a tube cell. And the smaller daughter cell is known as generative cell. Generative cell. Right? But this kind of division, which has resulted in two unequal cells, vegetative cell and generative cell, right? Because the division is a mitotic division, there is no change in the chromosome number of these two daughter cells to that of the parent microspore cell. So we know that microspores are haploid cells. Now, after the asymmetric mitotic division, it results in the production of two different daughter cells, and this cell is now no more considered to be a microspore. Now the cell is called as a pollen grain. So this differentiated cell, they are called as pollen grain, and a pollen grain is, as a microspore cell, a haploid cell. Alright, so in a pollen grain, because of the asymmetric mitotic division in the microspore, it has resulted in the production of two unequal cells. Right, and this asymmetric nature of the mitosis can be understood in terms of the difference in the content of the cytoplasm. Right, so there is an unequal division of the cytoplasm in these two daughter cells, even though they have received kind of an equal nuclear content, but the cytoplasmic content of these two daughter cells are very different. Vegetative cell is a larger cell because it has obtained more of the cytoplasmic content, whereas generative cell is a smaller cell because it has lesser of the cytoplasm. And because of which, vegetative cell is considered to be a very, very dense cell, having lots of nutrients and having lots of materials inside, which are required later for the development of the pollen grain. Right? And then we have the generative cells, which are usually the smaller cell, but they have a very significant role to play in the process of pollen development. Right? So anyway, what we understand here is, from a haploid microspore, what has happened is, at the first stage, we had the formation of a single large vacuole inside the pollen. Inside the microspore cell. And this has resulted in the pushing of the nucleus towards the wall of the microspore. And after this vacuolation, now we have the second event, which is called as an asymmetric pollen mitosis. And in this asymmetric pollen mitosis, because of the unequal division of the cytoplasm, the cell has become into two unequal daughter cells. Right? The larger cell is known as vegetative cell or tube cell, and the smaller cell is known as generative cell. And these two cells are collectively known as a pollen grain. Right? And the pollen grain is yet again a haploid cell. with no further change in the chromosome number. Right? So microspore is a haploid cell, pollen grain is a haploid cell, and such a haploid pollen grain cell having two of the cells, vegetative cell and generative cell, obtained through the process of a microspore maturation is technically referred to as male gametophyte. Right? So pollen grain is actually a male gametophyte. So we have to be very clear with this concept, right? Pollen grain is not a male gamete. Pollen grain is a male gametophyte. Alright, we learned about the two different life cycles in the plants. Right? So even in the angiosperms, we do have the sporophytic generation and the gametophytic generation. So sporophytic generation is basically about the formation of spores from the microsporangium. Right? Microsporangium gives rise to haploid spores by the process of meiosis, and that is the sporophytic generation. Now, the haploid spore on further maturation has resulted in a structure which is called as pollen grain, and pollen grain is a male gametophyte. It's not exactly the male gamete, it's a male gametophyte. And a male gametophyte is a structure or a plant body that would then give rise to the male gametes. So pollen grains are responsible for producing male gametes. Okay, but pollen grains are not male gametes. So please mark the difference here. Pollen grain is not same as male gametes. Pollen grains are the structures or the gametophyte body from which we obtain male gametes. And we are going to be learning about this in our further discussion. Alright, so this is the understanding about how we have the maturation taking place of the microspore inside the microsporangia, right? Or we can say these are all taking place inside the bilobed anther of an angiospermous flower. Okay, and even though we are learning all of these stages in terms of separate events, but we need to understand here that these events are all taking place simultaneously inside the anther, right? And it's a very rapid and a quick process. So after the maturation of the microspore into pollen grains, now we have a different reference to the sac. So earlier we called this as a microsporangial sac. Now, after having the pollen grains formed inside, it can be also called as pollen sacs. Okay, so the microsporangia are now the pollen sacs, and in the pollen sacs we have several thousands of pollen grains, which are all haploid cells, and these are the male gametophyte. So let's try to have a more detailed understanding about the structure of a pollen grain. So I have a structure of a mature pollen grain here. Now what we learned was that the microspore has resulted in the formation of two unequal cells because of the asymmetric pollen mitosis, and these cells were the vegetative cell and generative cell. So eventually what happens is the smaller cell, that is the generative cell, it loses the pollen starts losing the callose membrane, and as a result of which the generative cell gets engulfed inside, gets embedded inside the cytoplasm of vegetative cell. Right? So inside the cytoplasm of the vegetative cell you have the small spindle shaped generative cell. So these are the two cells of a mature pollen grain. Then externally, outside of the plasma membrane of the pollen grain, what we can also find is a maturing pollen grain would be having two of the wall layers. Right? So what I've shown here is I've shown the inner wall in a green sheet. Okay? And this inner wall is technically referred to as intine. And then outside of this wall, we have another thick wall which is called as exine. Right? So there are two wall layers even formed in the pollen grains. So the inner layer is the intine layer and the outer layer is the exine layer. Now the question is, where from these layers are originating? Okay, I'll just give you a brief understanding of this. So the inner intine layer, which is a more thinner layer, and it's a kind of a layer which is basically composed of cellulose and pectin. Alright, so such kind of the cellulosic layer is laid down by the microspore cytoplasm. So as the microspore starts maturing into a pollen grain, the cytoplasm of the microspore can be forming even these thin layers outside of the pollen grain, right? And that layer is the intine layer. Whereas this outer layer, the thick multi-layered outer layer, which is known as the exine, this is formed from the layer or the cell which surrounds the microsporangia, right? We learned about 4 different layers surrounding the microsporangia, and the innermost layer, which was tapetum, right? These tapetal cells are responsible for the synthesis of the outer layer, which is exine layer. An exine layer is a very, very thick layer, and this layer is very different from, in terms of its chemical composition as well, very different from the intine, because exine has a material which is called as sporopollenin. Alright, so exine is composed of a material which is called as sporopollenin. Now, why I want to stress with this sporopollenin in the exine is, sporopollenin is one of the most you can say resistant organic material in the plant, right? So sporopollenin is a material which cannot be digested by any enzymes, nor it can be degraded by any kind of an acid or any alkali or any such other organic substances, right? So this is considered to be one of the most resistant organic material known so far. And this material is present in exine, and because of which it provides with the basic, you can say, protection to a pollen grain when it has to be while it is maturing, or when the pollen grain is not germinating. So the pollen grain has to be stored for a long period of time, and in the storage of pollen grain we can be understanding that sporopollenin has a very significant role to play. So we know that exine is a thick layer. It is composed of a material which is called as sporopollenin, whereas the inner layer, as I said, intine, is composed of cellulose. So it is thin and it's basically made up of cellulose and pectin. So this is how we can be having a difference in terms of the 2 layers of pollen grain. And not only that, if we speak in terms of the exine layer, right, another difference is, if you look into the exine layer here, what you can be finding is that at certain region of the membrane, I have left a kind of a gap in between, right? So we know that exine is a very thick layer, but this thick layer is not a continuous layer, right? Rather it's more of a you can see a discontinuous layer having some pores in them, right? And what you can be seeing is I have shown 3 of the pores here. So we have a kind of a basic understanding that in cases of dicot plants, right? In the dicot plants, the pollen grains have usually 3 of these germ pores in the exine, right? Whereas in cases of the monocot plants, we can be expecting a single germ pore, right? That is how we have the basic understanding. So these openings or the apertures that we see in the exine layer are the germ pores. So this is a germ pore, which is an opening, or you can say an aperture, that is present in the exine layer. And in this region, this is an opening because, or this is a kind of an aperture because it lacks sporopollenin. Right? So in this region, we don't have any deposition of sporopollenin. Whereas if you look into the inner intine layer is a thin layer, and it is even a very smooth continuous layer. Right? So that is how we can be even having a way to differentiate between exine and intine. So these are the layers of the pollen grain. Right? Then the pollen grain can be having all different shapes and sizes. Right? So right now what you can be seeing here is I have drawn a more typical of a spherical pollen grain having some kind of you can say spiky or a spinous kind of surface, right? Irregular surface. Now some of the pollen grains can be having a smooth surface, some of the pollen grains can be having more of a rough surface. Now it depends on the plant species. And the pollen grains can also be of different shapes as well, right? It can be spherical, it can be elliptical, it can be reticulate. So we have all different shapes of pollen grains in different species, and we can also be expecting all different sizes of pollen grains. Right? Some of the pollen grains can be very small, like it can be around 5 micrometer in size, whereas some of the pollen grains can be more bigger, comparatively bigger. They can be around even 200 micrometers. Right? So this is how we understand with the structure of a typical mature pollen grain. Now this kind of a pollen grain, because we said there are 2 cells in this pollen grain, right, the larger vegetative cell and the smaller generative cell, such a pollen grain is said to be a bicellular pollen grain. Right? So this is a bicellular pollen grain. A bicellular pollen grain because it has 2 cells inside. Bicellular pollen grain. So what do we mean by bicellular pollen grain? It is having 2 cells. And we know that the cells are even different in their sizes, right? So most of the pollen grains are usually, you can say, released from the anther during this stage, right? The stage which is called as bicellular stage or bicelled stage of the pollen grain, where we can be anticipating 2 of the cells inside, vegetative and generative cell. Right? But in some of the pollen grains, right? In some of the species, we can say the pollens might even be released at yet another stage, which is called as a tricellular pollen grain. So the pollen grain can be released either in a bicellular stage, or it can be even released in a tricellular stage. Now, what is this tricellular stage of a pollen grain? So tricellular, which means to say we have to be expecting 3 cells in the pollen grain. So such kind of a tricellular pollen grain is formed when we have a further mitotic division in the generative cell. Right? Vegetative cell is the large cell which is not basically involved in the making of the gametes. That is why we call the cell. The name itself is stating it is a vegetative cell, non-reproductive cell. Whereas this generative cell, this is a reproductive cell. Right? Actually a reproductive cell. And this generative cell can undergo a second mitosis. Right? Already it had underwent the first mitosis forming 2 of these cells, and then the generative cell in the pollen grain can undergo a mitosis, a single mitosis, and give rise to 2 male gametes. Right? The two daughter cells are now said to be 2 male gametes. And if we have a vegetative cell along with 2 male gametes from the generative cell, then it becomes tricellular pollen grain. So tricellular pollen grain having 3 cells in it. And which are these 3 cells? So we can be having 1 vegetative cell, and then we can have 2 male gametes. So if a pollen grain is released at this stage of the cell, that is the tricellular stage, right, then it is considered to be 3-cell stage of pollen grain. And in most of the angiospermous plants, over 60% of the plant species, we have the pollen released during this bicell stage, right? Whereas in a few of the other species, we can be even expecting the pollen to be released when the pollen already had made the male gametes while it is still inside the anther, right? But in most of the cases, they are released in this bicell stage, which means it is just the vegetative and the generative cell. And then the generative cell, once the pollen grain lands on the stigma of a flower, then it starts germinating, it starts producing the pollen tube. And once the pollen tube is close to the micropylar end, we'll understand later, at that time then the generative cells can be, so in the pollen tube the generative cells can be then undergoing the mitotic division, resulting in the production of 2 male gametes, right? So such is the case if it is a bicelled pollen grain, where the generative cell doesn't divide while it is in the anther, but it divides when it's in the pollen tube, right? And that is the bicelled pollen grain. So in a tricellular pollen grain, the division of the generative cell had already completed when it is in the anther, right? And had already produced the 2 male gametes, which are ready for double fertilization. So this is the understanding about the structure of a typical bicellular pollen grain. Now if you go in terms of the functions of these 2 cells, right? So one already we have had discussed here in terms of the function of the generative cell, right? So generative cell functionally is very important because it helps in the formation or the production of 2 male gametes, right? So it produces male gametes. So this is actually the one which produces male gametes, right, as we have discussed, right? Then the vegetative cell. Now, as I said, these are vegetative because they are not forming the gametes, male gametes, but then these are vegetative cell, or even they are referred to as tube cell because they are helpful in terms of the formation or the synthesis of pollen tube. So this produces pollen tube. So that is how we can say that both of these cells have a kind of, you can say, important function to play as the pollen has to be maturing and then as the pollen has to be germinating onto a female body. Okay, so the vegetative cell is vegetative because it's not producing any gamete, but then it's a vegetative cell which can be forming the pollen tube once the pollen grain lands on the compatible stigma. And such kind of the function is taken up by the vegetative cell, whereas the generative cell is responsible for producing 2 of the male gametes. And these 2 male gametes are produced by a single mitosis. Right? So by a single mitosis, generative cells can be producing 2 daughter cells, and these 2 daughter cells are now called as male gametes. And that's the reason why. That's the reason why we call pollen grains as a male gametophyte. Right? So this is a male gametophyte which can, right, to be very precise, it is the generative cell which produces the male gametes. So because the pollen grain can produce the male gamete, they become the male gametophyte, right? And the male gametes are actually produced from generative cells division. So generative cell undergoes a single mitosis giving rise to the 2 male gametes. And this kind of the division, mitotic division of the generative cell can be initiated when the pollen grain is right inside the anther itself, right? If that is the nature of, you can say, the initiation of mitosis, then it becomes tricellular pollen grain, which occurs in you can say some of the angiospermous plants, whereas in over 60% of the angiospermous species, we understand that the mitotic division in the generative cell occurs in pollen tube, right? And the pollens are released at the bicellular stage. Okay, so that's how we can be basically keeping up with our understanding about the structure of a typical, you can say, bicellular pollen grain. Next, as the pollen grains mature, anther starts to prepare for the dispersal of these pollen grains, and this process is anther dehiscence. So what I have written up here is anther dehiscence is the splitting open of anther wall along the specific, you can say, region in the anther, and this region is what we call as the line of dehiscence, because this is a region where we have the inbuilt weaker cells, right? And because of which anthers can be easily opening up from such area. And the line of dehiscence usually lies in the indentation region, right? The depressed region in the anther lobe. So during this anther dehiscence, what happens is the outermost layer of the anther, right? We know that anther has 4 layers. So the outermost layer, which are the epidermal layer. So these epidermal layer, especially around the indentation, has a kind of a modification, right? These cells are eventually going to be getting dehydrated, and as a result of which they are going to be getting degenerated, right? And these epidermal cells are considered to be stomium. So we have stomium at this indented line of the dehiscence region, which gets dehydrated and which gets degenerated. And not only that, the inner layer to the epidermis, the layer beneath the epidermis, which is endothecium. So that endothecial layer even gets a kind of a secondary wall deposition. So secondary wall deposition is basically we have more of the thickening, right? We have more of the lignification taking place in the endothecial layer. And this kind of the deposition, the thickening which occurs in the endothecial layer, even is a kind of not a uniform thickening, right? We have a U-shaped thickening. The inside wall of the endothelial cells get more of the deposition of the lignin, whereas the outer layer or the outer region, right, which is facing towards the stomium, so this region of the endothelial cell will not be getting much of the lignin deposition, and as a result of which it creates a kind of a force. And that force would be causing a pressure which pushes the anther wall outward, right? So it pushes the anther wall outward, and as a result of which we have the anther wall opening, right? So in this way, in this simple way, we can be understanding with the anther dehiscence. So basically we say we have epidermal cells. So the epidermal cells, especially, right, across these region, that is the line of dehiscence, which are called as stomium. So these epidermal cells would be undergoing a kind of dehydration and degeneration. Okay, so undergoes, let us see, dehydration. It loses water, it dries up, okay, and would eventually degenerate, right? So that is one of the ways through which we can be having the preparation for dehiscence. And secondly, we can talk about the secondary thickening, right? So we have secondary thickening. And this secondary thickening is actually technically referred to as lignification, because we have more of the lignin deposition taking place. All right. So secondary thickening or lignification of which layer? The layer beneath the epidermis, which is endothelium. So we have secondary thickening of endothelial layer. right? And that kind of the secondary thickening, lignification of the endothelial layer, which is going to be not very uniform thickening, is going to be pushing the, so it pushes, right? Which would be helping in, it creates a force and it pushes the cells to go or to bend outward, and that results in the opening of anther. And this is how we have the dispersal or the release of pollen grains from a mature anther. And as we said earlier, this kind of the dehiscence can be occurring either you can say during the 2-cell stage of a pollen grain, or it can be taking place when the pollen grain is already in the 3-celled stage. After the release of the pollen grains from the anther into the external environment, now the pollen grains has to be remaining viable, which means to say the pollen grains has to be having the ability to germinate and produce male gametes and thereby cause fertilization once it lands on the stigma. And such kind of the viability of the pollen grains varies from species to species. So if I have to take an example from your textbook, for example, in rice and wheat, the viability of the pollen grain is a little shorter. They can be remaining viable for around 30 minutes after its release from the anther. Whereas in cases of some of the legumes, the pollen grains can be remaining viable even for months. So such kind of the viability would be affected by several factors. Right? It could be the physical factors, external physical factors, like it can be the temperature or humidity, or it can be the internal factors as well, right? Basing on the nature or the deposition of its wall layer. So we learned that in a pollen grain is well protected by an outer exine layer made up of sporopollen. So sporopollen being a very the most resistant material. It can also be withstanding the enzymes and the acids that are there inside the digestive system of a browsing animal, and as a result of which, even if a pollen grain is eaten up by an animal, it can still be remaining viable and it can still be causing germination. So in order to be keeping the pollen grains viable for a long period of time, we also have certain, you can say, man-made techniques, right? We have the maintenance of the pollen grains. We have the storage of pollen grains, right? And such kind of the storage is done in pollen banks, right? These are done by various centers and institutes where they keep or they maintain the different pollen grains. And these storage of these collection of the pollen grains are known as pollen banks. Okay, so we also have many centers in India who are maintaining such pollen banks. To give you one of the examples, we have a center located in Bangalore, which is the Indian Institute of Horticultural Research. So this is also an active center where we have the storage of pollens. And in these banks, they can be storing the pollens through different methods. It could be a short-term method or it can be a long-term method. And in the long-term method, one of the very, you can say, effective methods is a method that is called as cryopreservation. So in this method of cryopreservation, the pollen grains are stored in an extremely low temperature. That is, they are stored in liquid nitrogen. Okay, so cryopreservation, we have pollen stored in liquid nitrogen, which means to say it is stored in a temperature which is minus 196 degrees Celsius. So such as you can say an extreme freezing temperature in which we have the storage of pollen grains, and cryopreservations and many other methods of, you can say, storing the pollen grains would be helping in the preservation of biodiversity, as well as these can be helping in you can say many of the different the pollens can be later used for many different plant breeding programs. Okay, now if you're interested to know more about this process, which is cryopreservation, what I want to be sharing with you is now in the coming nearing few days, that is on the 5th of June. 5th of June being the World Environment Day. So the Indian Institute of Horticultural Research located at Bangalore, they are going to be holding a webinar. I'll share the link with you. So if you're interested, you can even register and participate in this webinar. So they're going to be holding a webinar on 5th of June and they have a series of program in which one of the presentation would be, or the the discussion would be on cryopreservation. Okay, so cryopreservation, to just give you a kind of a simple understanding, cryopreservation is how we have the storage of here in this case the pollen grains in extreme low temperature, that is in the liquid nitrogen at minus 196 degrees Celsius. Okay, so this is how we can be also understanding that pollen grains basically needs to be surviving in an environment which could be sometimes getting harsh, and during such harsh environmental conditions, pollen grains have their own, you can say, thick outer layer, which is the sporopollenin, which can keep it viable for a certain period of time, as well as we can say we have human intervention by which we are able to be preserving the desirable pollen grains, or we are able to be preserving the pollen grains in order to be keeping up or conserving the biodiversity. Finally, let's look at the advantage and disadvantage of pollen grains. So pollen grains are edible, and they are considered to be rich in carbohydrates, proteins, vitamins and many other essential nutrients, and because of which we have pollen supplements in the markets which are available in the form of tablets and syrups. And these pollen supplements are thought to be very useful to athletes as well as to young children. But at the same time, pollens are considered to be a major cause for allergy. Right? Allergy is a kind of, you can say, a hypersensitive response by our immune system whenever we come across with certain unsuitable, let's say, agents, right? So pollens are considered to be one of the major cause for allergy, and this can be resulting in a condition in some of the humans called as hay fever. So hay fever is basically a condition in which you will be developing a cold-like symptom, right? Where you will be facing a runny nose, you will be having sneezing or watery eyes or itchiness or inflammation. So all these allergic responses could be also caused by pollens. And one of such plants, or the pollens which are causing hay fever in many individuals, is a plant known as parthenium. So parthenium is a very common wheat that grows everywhere, and the pollen of this plant is considered to be mainly responsible for causing hay fever in many individuals. And some of us even have a kind of a reference to parthenium as asthma plant, right? So that's the reason why we should be making sure that if an individual has a kind of allergic response, so we have to avoid the allergen. Particularly, we can be plucking off the parthenium if it is the cause for your allergy. So with this, we come to the end of today's session, which is on the formation of the male gametophyte or pollen grain, and we also discussed with the structure and various other aspects of pollen grains. Here is the screenshot of the upcoming webinar which I mentioned earlier. So you can go through the program details, and for those of you who wish to take part in it, you can go to the link which I've shared in the description, and get yourself registered before it gets full. And these are the assignment questions for the week. There are 8 questions. Well, that's it. Till then, take care.